So welcome everyone to the Metrics International Forum. The forum is organized by the Meta Research Innovation Center at Stanford, or short Metrics. The center is directed by John Ioannidis and Steve Goodman, and we are a research group and fellowship program dedicated to finding ways to improve the validity and transparency of scientific research. I'm Mick Siebert, a postdoc at Metrics, and I will be today's moderator. All of our international forums are recorded and can be accessed on our website. Please keep your questions until the end or write them in the chat during the presentation. Our presenter today is Mauricia Davidson. She is in her final year of doctoral studies at the Université Paris-Cité, working under the supervision of Isabelle Boutron and Anna Khaimani. She has a keen interest in epidemiology, research methodology, and interdisciplinary science. Throughout her academic journey, she has demonstrated a strong command of evidence synthesis, specializing in living systematic reviews, notably contributing to the COVID-NMA initiative. She's currently investigating the integration of various types of information in living systematic reviews, focusing on preprints, post-publication peer review, and observational studies. So if you're ready, Mauricia, we can start.
Yes, well, thanks, Max, for the introduction. Good morning, everyone, or good afternoon. Actually, as some of you start your day, I'm ending mine as it's 5 p.m. here in Paris. But anyway, I'm happy to be here to discuss the comparison of effect estimates between preprints and peer-reviewed journal articles of COVID-19 trials. So preprints, what do we know about them? They're the non-peer-reviewed version of a scientific article submitted to publicly accessible platforms. And why? Because peer review is slow. The traditional peer review process often requires several months, which can delay the implementation of effective treatments in clinical practice. The example that I've given here is actually from the recovery dexamethasone trial, whose full peer-reviewed paper was published online 1 month after its preprint. And in that time, over 700,000 new COVID-19 cases were confirmed. So one can even say, and actually the lead author of the recovery trial even said that waiting for peer review could have undoubtedly cost lives. However, the lack of peer review raises concerns regarding the reliability of the preprint results. So for example, back in January 2020, a bioRxiv preprint suggested that the coronavirus had similarities to HIV. Of course, it was swiftly criticized and withdrawn within 2 days. So, and other questionable preprints that turned out to be fraudulent actually were subsequently retracted include a study posted to SSRN and a Research Square RCT claiming that ivermectin reduces COVID-19 deaths. So as the debate on preprint use and the inclusion in evidence synthesis continued, we decided to take a look at our database, so the COVID-NMA database, which contained a good portion of COVID-19 treatment RCTs, first available as preprints and also available as peer-reviewed journal articles. And in doing so, we aimed to evaluate whether effect estimates differ between preprint and peer-reviewed journal articles. So for those who are unfamiliar with our database, the COVID-NMA Living Systematic Review aimed to provide decision makers with up-to-date, complete high-quality data synthesis on interventions for the prevention and treatment of COVID-19. It relied on a complex process whereby every working day we searched electronic databases, particularly the LOVE platform and the COVID-19 study register. And as soon as a new trial was identified, data extraction and risk of bias assessment were done in duplicate, followed by analysis and grading of the evidence once per week. Now, later on, we changed the process so that meta-analysis was done bi-weekly, but all our data is on the COVID-NMA platform and was used by guideline developers and the WHO. So in a first step, we conducted a meta-epidemiological study. So generally, meta-epidemiological studies use collections of meta-analyses to investigate the the link between a trial characteristic and the effect estimate. So in this case, the trial characteristic was publication type, so preprint or journal article. And at the point of evidence synthesis, it is, so why we chose to do this, because at the point of evidence synthesis, it is unclear which preprints will eventually be published in peer-reviewed journals. And of course, in our evidence synthesis, in our database, we included both preprints and peer-reviewed articles, and we meta-analyzed both. So therefore, by examining meta-analyses themselves via this type of study, via meta-epidemiological study, focusing on those that include different trials of different publication types and estimating checking to see if there was a statistical difference, we can better assess the reliability of preprint results. So that was our idea going in and why we chose to do a meta-epidemiological study. So we searched the COVID-NMA database on July 20th, 2022, and we included all meta-analyses of pharmacological treatments versus standard of care or placebo for patients with COVID-19. And of course, these meta-analyses must include at least one preprint article and one peer-reviewed journal article. We also considered the COVID-NMA defined critical outcomes like clinical improvement at day 28. Clinical improvement was defined in the COVID-NMA protocol as an improvement of at least 2 points on the clinical progression scale or hospital discharge. We also considered WHO clinical progression score of level 7 or above, which essentially equates to intubation or death, all-cause mortality, adverse events, and serious adverse events. And we excluded meta-analyses of preventive interventions, vaccines, and non-pharmacological treatments, and RCTs that reported 0 events in both study groups, in the intervention and comparator group. So I briefly touched on the COVID-NMA process earlier, but imagine this is the database. So this is the COVID-NMA database with the different meta-analyses in orange. So as part of our COVID-NMA protocol, we meta-analyze data regardless of the publication type. And so when we identified a new RCT report, we extracted the data and analyzed weekly and then biweekly, and then saved the database. So imagine these are the saved versions of the database containing the multiple meta-analyses at different time points from inception of the COVID-NMA initiative in early 2020. And so weekly, we saved it until, so we saved it, and these are the saved databases from inception to our search date for our project in July 2022. And so meta-analyses were selected for inclusion in our final data set in 2 steps. So first, using R, we randomly selected a time point where a meta-analysis included at least one preprint article and one peer-reviewed journal article, right? So secondly, if at this given time point, a meta-analysis meeting the eligibility criteria was available for more than one outcome, the meta-analysis that we included was the one with the highest number of trials. Was the one with the highest number of trials. We selected that meta-analysis. So now in order to estimate the difference in the effect estimate between preprint and peer-reviewed journal RCT, we calculated the ratio of odds ratios. So whereby an ROR of less than 1 indicated that preprint trials yielded larger effect estimates than journal articles. So here is our data. We identified, well, here's how we got the data. We identified 323 meta-analyses of pharmacological treatments in COVID-NMA database. we excluded those that compared two active treatments and performing the random sampling to include at least 2 RCTs of different publication types for critical outcome, and RCTs with no events in both groups. And in the end, we had 37 meta-analyses that included 44 preprints and 70 journal articles. So some characteristics of our study sample was that the sample size, the median sample size was 199 participants. The median number of RCTs per meta-analysis was 2. So it was a small amount actually. The majority of the trials were prospectively registered, had received industry or mixed funding, and they were multicentric trials that were rated, the majority were rated some concerns of risk of bias. So here is the data. So the forest plot depicts our final analysis. So these are the 37 meta-analyses. So the treatment comparisons versus standard care or control, standard care or placebo, are the 37 meta-analyses, and these are the specific outcomes that they evaluated. So all-cause mortality, adverse events, WHO score 7, etc. And for each specific analysis, meta-analysis, these are the number of preprints or the number of journal articles included in the meta-analysis with their respective RORs. So the pooled ROR was 0.88 with an insignificant confidence interval, suggesting no evidence of an association between preprint and peer-reviewed journal article on the treatment effect estimates. But considering the small number of trials within most meta-analyses and the large uncertainty of the respective RORs, like, for example, this one, insanely very large confidence interval. So based on these factors, we couldn't draw any strong conclusions. So that was the primary analysis. So the 0.88 was a primary analysis, which included at least one preprint and at least one journal article. As you can see, some of them also had more than just one. So it just had to be at least one for both publication types. But we also conducted post hoc sensitivity analyses to examine the impact of early synthesis. So whether early on, when we only have 2 trials in the meta-analysis, so we have exactly one preprint and exactly one journal article. And then we also did it where we are trying to look at synthesis at later stages. So more than 3 trials in the meta-analysis. And of course, this was all done, once again, via random sampling and ensuring heterogeneity in publication type, as I said, and we found similar results. So that was the meta-epidemiological study. But in a second step, we looked at a one-to-one, like a direct one-on-one comparison of effect estimates from preprints and from journal articles. So we utilized the same COVID-NMA database, searched on July 20th, 2022, and we also looked at RCTs of pharmacological treatments and on the same COVID-NMA defined critical outcomes. In this study, however, we specifically included RCTs that were first reported as a preprint and subsequently as a journal article. So it was the same RCT, but they had to have both a preprint and a journal article. And we ensured that they reported the same analysis in both documents. So meaning that we excluded RCTs that reported an interim analysis in the preprint and a final analysis in the journal article. So we systematically explored whether the number of events, the number of participants analyzed, and the treatment effect estimates were consistent. So we did this by looking at discrepancies in results reported in preprints and subsequent journal articles. And these discrepancies were either a change in the effect estimates of at least one outcome. We also were looking for a change in the direction of the effect estimate, a change in the statistical significance, change in overall conclusion. And we also looked at whether the outcomes were added or deleted in the journal article compared to the preprint. So what we found was 135 preprint and subsequent journal article pairs of RCTs of RCT reports in the COVID-NMA database, and after excluding based on our eligibility criteria, in our analysis for this study, we had 109 preprints that were subsequently published in peer-reviewed journals. So some general characteristics: the median sample size was 150 participants. The median delay between the first preprint post, the first version of the preprint, because some preprints, lots of preprints have multiple versions. So we specifically looked at the first version. So from the first version to the peer-reviewed publication, we had, there was a delay of 121 days, a median delay. Once again, majority of the trials were prospectively registered, received industry or mixed funding, were multicentric trials, and were rated as some concerns of risk of bias. So here are some characteristics. We found that actually in 81 trials, all of the outcomes, all of the data, all of the effect estimates were consistent for all outcomes in 81 trials. In 28, we had some discrepancies, namely 9 of them actually had a change in the effect estimate between preprint and journal article. So these are the 9 trials and the outcomes that were specifically had specific changes. As we can see, the effect estimate was generally similar. There were small changes, mainly due to either a change in the actual number of the events of the outcome or a change in the number analyzed, but these were typically small changes. For example, here we have 26 versus 20, 26 versus 27, or 28 versus 29. So basically what we found was 135 preprint and journal article pairs of RCTs reports in the COVID-NMA database. And after we excluded, based on our eligibility criteria, we ended up with 109 preprint journal article RCTs in our analysis. So here are some of the data, the general characteristics. Median sample size was 150 participants. Median delay between first preprint, post, and journal article publication was 121 days. The majority of trials were prospectively registered, had received industry or mixed funding, were multicentric trials, and were rated as some concerns of risk of bias. So this is what we found. We got that 81 RCTs had had completely consistent data between its preprint and its subsequent peer-reviewed publication in all outcomes. That's for 81 RCTs. For 28 RCTs, there were some changes. Namely, 9 of them had changes in the value of the effect estimate. So this forest plot shows some of the changes in the effect estimate, shows the changes in the effect estimate, and we can see that they were quite small changes, namely due to either a change in the actual number of events, of the outcome events, or the number of participants analyzed. So for example, here we had 26 to 27, 28, 29, and most of these were due to data cleaning data cleaning. We had one trial that had a change in the direction of the effect estimate, and but no trial had a change in the statistical significance. There was no change in the overall conclusion. We had 14 RCTs that had an added outcome in the journal article versus the preprint. 6 had a missing outcome in the journal article versus the preprint. So what we also tried to look at was to see if there was any difference between published preprints and unpublished preprint, because the forest plots and everything showed for preprints that obviously were subsequently published. But let's just take a look at what are some of the general characteristics of the unpublished preprints, because actually we cannot really determine um, if these preprints were not published because of unsupported conclusions. So we can't actually do a direct comparison for the unpublished ones, but just in terms of general characteristics, we found that they were generally similar. We saw similar numbers between unpublished and published preprints. Now, of course, we have some limitations of our study. First of all, we focused on COVID-19 trials, which may not be representative of preprints and journal articles in other fields outside of the pandemic. Of course, we know that peer review was majorly affected by COVID-19, with significant fast tracking of the process and difficulties accessing highly skilled peer reviewers. Secondly, our study was also limited to the decisions of the living review. For example, protocol revisions could affect the sample composition. And we considered only RCTs, so we meta-analyses of RCTs as well. So, and RCTs rely on pre-registered protocols, so results could be different in other study types like observational studies. Now, particularly regarding the meta-epidemiological study, the number of RCTs per meta-analysis was small, with a median of 2 RCTs per meta-analysis, which increased the uncertainty around the estimation. So we could not account for other things that we were planning to do, particularly account for pre-specified covariates in a meta-regression analysis, and so we can only rely on subgroup analyses due to the small number of trials. And regarding the second study, for those preprints, as I said, that were never published in a journal, we could not evaluate whether peer review prevented journal publication due to unsupported conclusions. But we found that the trial characteristics were generally similar between the published and unpublished preprints. Now, what can we draw overall from this is that what we found is that there was no evidence of important difference between treatment effects between preprint and peer-reviewed journal articles. But the results should be interpreted with caution due to small number of trials within most meta-analyses and the large uncertainty around the respective RORs. We also, from the second study, we found that effect estimates were generally consistent between COVID-19 preprints and subsequent journal articles. The main results and interpretation did not change in any trial, but some outcomes were added and deleted. So what can be implied is that within the constantly evolving research landscape, and especially in the context of a pandemic, considering the results of preprint trials may be reasonable. But systematic reviewers and meta-analysts should assess preprint inclusion individually, of course, accounting for risk of bias and completeness of reporting. Thank you very much, and I look forward to your questions.